In this lab, we'll be providing an overview of the regions of the brain. The regions we're going to cover are going to include the cerebrum, which is the highest part, the largest part you'll see. Just below that will be the diencephalon. Anything that has the name thalamus, we're going to see is located in the diencephalon, the thalamus, the hypothalamus, the epithalamus. There's even a subthalamus. We're going to look at the thalamus, the hypothalamus, and the epithalamus today. Below the diencephalon, we'll study the brainstem, kind of the primitive brain, which includes the midbrain, the pons, and then the medulla oblongata, which is connected to the spinal cord. And then we'll finish off with the cerebellum. We'll also take a look at the ventricular system, a system of ventricles and canals that make and contain cerebrospinal fluid. We'll finish off the lab with the ventricular system. So let's begin this lab with looking at the cerebrum. A couple of definitions I need you to know and be able to identify on our models. There's so many neurons in the brain. In order to get them to fit into the cranial vault, the brain has to kind of be, scrunched together to fit in there. And when we do that, we actually end up getting these outward folds and these inward folds that we can identify. So I took a photo of the brain. This is a superior view. We're looking down on the brain. And if you take a look, all of these outward folds here, they're kind of directed outward. They kind of look like worms, almost kind of wormy. All of this, you follow my cursor around, these are known as a gyrus, right? Singular would be gyrus, gyri would be plural. If you take a look between my cursor now here on the right and then here on the left, these are two gyri. There's a fold that goes in between the two gyri. That is known as a sulcus. And so a sulcus you can think of as a shallow groove. Right? A gyrus is an outward fold. A sulcus is a shallow groove. The next two definitions are the cerebral cortex and the cerebral white matter, which consists of tracts that we'll study in lecture, as a matter of fact. Cortex. A cortex is an outer surface, a covering. The term we use a lot for a lot of different organs when something has an outer layer and an inner layer. So think of the cerebral cortex as the outer layer, and it is primarily made up of gray matter that's superficial on the cerebrum. Again, from our previous lab on the spinal cord, we had learned that gray matter is anything that's unmyelinated tissue. Anything that doesn't have myelin will appear gray. Underneath the cortex is the cerebral white matter. So I took a brain and we just kind of sliced it and took a look in here. You follow, it's maybe 4 millimeters thick on a real brain. This thin superficial layer here, it's kind of painted in green on this model. All of this is the cerebral cortex. And then if you look deep to it here, this is all the white matter consisting of tracts that are just sending information to different parts of the brain and down to the spinal cord. We can also see on this section here, this outward fold would be a gyrus. And then right above my pointer here, this would be a sulcus. So we are able to see that also. So again, if you were doing dissection and you took a brain out, you were just holding the brain on the outside, you would be touching the cerebral cortex. And then we would have to cut into the brain to see the white matter tracts below. The next definition, our brains are divided into a left and right hemisphere, right? Hemisphere meaning just a half a brain, hemi meaning a half, a sphere is a circle. And that hemisphere, the hemispheres are divided by a deep groove known as a fissure. So we said earlier, a sulcus is a shallow groove, a fissure is a deep groove. The fissure that actually separates the left cerebral hemisphere from the right cerebral hemisphere is known as the longitudinal fissure. So here I have the brain, and we're able to see this would be the frontal area, this would be the posterior aspect over here. This deep groove separating the left and the right fissures is going to be the left and right hemisphere is known as the longitudinal fissure. Projecting down into the longitudinal fissure is an extension of the dura mater. It actually goes down into the the fissure and acts as the stabilizer of the brain. This structure is known as the falx cerebri. So this is actually an extension of the dura mater, which is one of the meninges, the outer meninges, and it projects down into the longitudinal fissure. I included a couple blood vessels. These are important for AMP2 when you study how the venous drainage occurs in the brain. You'll actually look at these large veins known as sinuses. This big guy right up here at the top kind of runs underneath the parietal bones, frontal bone, occipital bone. This is known as the superior sagittal sinus. The smaller line over here is known as the inferior sagittal sinus. This one going across over this way is the straight sinus, and then we can see a little bit of the transverse sinus. So I just wanted to introduce some of those terms. You'll study those in AMP2 in much more detail and see how the brain is drained of the venous blood and cerebrospinal fluid. Back to our notes. So we have the longitudinal fissure. Within the longitudinal fissure, you'll find the falx cerebri. One other important fissure that we want to take a look at. The cerebrum in the back has the occiput. I take it back. The cerebrum in the back has the cerebellum located below it. So this is a posterior view of the cerebrum. So we can take a look all back here. This is what we're going to see in a couple of minutes. It's called the occipital lobe. And below the occipital lobe, this structure here, we'll see the left cerebellum. Over here is the right cerebellum. There's a groove that runs across, a deep groove separating the occipital lobe above from the cerebellum below. That lobe, or that groove, we're going to call the transverse fissure, the transverse fissure. And we know that transverse means across, so the fissure runs across. This fissure also has a extension of the meninges running in there, also known as the tentorium cerebelli. So same as the longitudinal fissure had the falx cerebri, the transverse fissure contains an extension of the dura mater known as the tentorium cerebelli. So if I go back to this model over here, if you recall, this is the falx cerebri. This would sit in the longitudinal fissure. Down over here, this and this over here, this would actually slip into the into that transverse fissure, and this is known as the tentorium cerebelli. I have it here just by itself. So this is the tentorium cerebelli. Tentorium means to tent or to cover. Cerebelli would be the cerebellum. So this is actually on top of the cerebellum, and it helps to kind of keep the occipital lobe off of the cerebellum. The next couple definitions. There are a couple shallow grooves that we need to know. One really important one. A lot of clinical things happen in this region. It's known as the central sulcus. Roughly in the middle of the brain, kind of coming up the left side of the brain, going over the top and going down the right side of the brain, there is a shallow groove that we're going to call the central sulcus. So on this photo, I just took a top picture, and we can see, again, this would be the frontal area. We'll learn that in a minute. This would be the posterior aspect back here. If we follow my cursor, coming up the left side here, there's a shallow groove kind of between the red and the blue. And then on this side, it's not color-coded, but we'll see this groove going down the other side over here. This is known as the central sulcus. So again, look for it in the middle of the brain. Even if it's not a color-coded brain, you'd be able to find it very, very easy because it's continuous. It just kind of comes up one side of the brain and then goes down the other. Here I took a side view, and we can see the central sulcus again, just kind of going up, in this case, the left side of the brain, and then it would go over the brain and down the other side. The other sulcus I'd like you to know is right over here. This is called the lateral sulcus. The lateral sulcus is above this lobe here, which we're going to learn in a minute, is called the parietal lobe. And this lobe here in the front, the red one, is all going to be the frontal lobe. And this lobe down over here is called the temporal lobe. So the lateral sulcus is a shallow groove relatively that goes between the two lobes above, and the temporal lobe below. The brain gives rise to 12 cranial nerves. The cranial nerves that we're going to know for this lab include the first cranial nerve number 1, and the second cranial nerve number 2. The other cranial nerves will go over 3 through 12 as far as their function, but these we're actually going to have you identify on our models. So cranial nerve number 1 is the olfactory nerve. Olfaction means to smell. So we actually find these nerves right over the nasal cavity. Cranial nerve number 2 is the optic nerve. These nerves go right back to the back of the eye. They enter the eye, and they're going to be stimulated by the cells in the retina when they're hit by light. So let's take a look at where they are. So on this picture here, I took our brain and I turned it upside down. So we're looking at over here, this is the frontal area, the anterior aspect, and down over here below, we're kind of showing you the posterior aspect. These two structures right here, this would be the right olfactory nerve. This would be the left olfactory nerve. Again, they're located right over the nasal cavity, and they send fibers down, if you recall, through the cribriform plate in the ethmoid bone, and they're responsible for, these two nerves are responsible for smell. The two parts of cranial nerve number 1 we'd like you to know, at the end of the nerve, there's a little bit of a swelling. This is known as the olfactory bulb. From there, the fibers will go this way, posterior, and this will be called the olfactory tract. Cranial nerve number 2 has been cut right here. This is going to be the one that's on the left side, and this is cranial nerve number 2 on the right side. This nerve would actually extend over to here, and the eyeball would be like right about here, and this cranial nerve would extend over this way, and our eyeball would be somewhere over here. So the eyeballs obviously have been taken out, so the cranial nerves are cut. So they do go to the posterior aspect of the eyeball. What's interesting is the, say, cranial nerve number 2 coming from the right eye over here, cranial nerve number 2, as it extends back into the brain, crosses over to the left side of the brain. So you can see right over here, this actually crosses. Same thing with cranial nerve number 2 on the left. It crosses over right over here. So this region where there's a crossing is known as the optic chiasm. Okay, so you should be able to identify that crossing. That's the cranial nerve number 2, the optic chiasm, and also know that cranial nerve number 2 is for sight. Our cerebrum has 5 lobes that I'd like you to know. For lab, I do want you to be able to identify. For lecture, I do want you to know their function. So let's take a look at the frontal lobe first. Frontal lobe does a lot of important things for us. It controls higher psychic functions. Our personality comes from there. Our planning, right? You're planning on maybe going to nursing school. You're planning on getting married, that type of thing. Concentrating, right? When we concentrate. And then also something very important, all of our voluntary movement originates from the frontal lobe. So everything that you do voluntarily to contract your muscles is going to be originating from the frontal lobe. There's an area on the frontal lobe I'm going to show you called the precentral gyrus. So let's talk about the words for a minute. Pre means before. Central would be where that central sulcus is, and we know from our first slide that the gyrus is an outward fold. The reason this precentral gyrus is important is that it contains what we call the primary motor cortex. All of our voluntary movement originates there. So I took a picture, again, of the lateral view. So guys, here was the central sulcus coming up over here. Everything anterior this way is the frontal lobe. This gyrus right here in front of the central sulcus, directly in front of the central sulcus, that is the precentral gyrus, also known as the primary motor area. So again, all of our voluntary motor movements originate here, and it's all spatially related. This area here would kind of control the hip area, and they all control different areas right here. So if somebody were to have a stroke, or some type of trauma to this gyrus, this would actually cause them to have some type of paralysis probably, or some type of weakness in their muscles. Right? So a lot of clinical situations can happen over here. All right, so frontal lobe, again, higher psychic function, personality, planning, concentration. It contains the precentral gyrus, which is important. There's an area just, let's see, we have this just anterior, right over here, labeled it for you. This is called the somatic association area. Here, this is where over the course of your life, where you're learning to do movements and practice movements, like playing the piano or writing and that type of thing. They call this an association area, and this is where you kind of learn those types of movements. But ultimately, everything comes from the precentral gyrus. This is where our primary motor movement area is. Go back over here. The second lobe that I'd like you to know is more of a sensory lobe. The frontal lobe is more of a motor lobe. The parietal lobe is more of a sensory lobe. It contains a gyrus that I also need you to know that's very important, called the general sensory area. It's located on the postcentral gyrus. So all the nerve impulses that are coming up from our body, sensing various modalities, things like pain, pressure, touch, vibration, hot, cold, all of these sensations ultimately make their way up to the parietal lobe, where you perceive them and you sense what you're feeling coming from your body. So the postcentral gyrus on the parietal lobe is what is going to sense that. So again, here was the central sulcus. And then if you go directly behind it, at this blue outward fold here, this is all the postcentral gyrus. This is known as the primary sensory area. This is where you sense everything on your body. Similar to the precentral gyrus, this would be like the hip area here of the lower extremity. So again, if you just kind of touch your hip on the side of your, with your hand, just kind of touch there, this is where you would actually perceive it. The somatic sensory area, this is where the impulses that are brought up to the primary motor area, the primary sensory area, are given some type of meaning. So if you touch your hip, this would be like where you actually realize, oh, it's me, I'm touching my hip, I'm trying to understand this lesson by touching my hip, that type of thing. You're actually applying some type of meaning to the sensation that's coming up. And so the primary sensory area is where you actually sense it, and this actually gives it some type of meaning. Okay, so the parietal lobe, again, this slide's an important slide. The frontal lobe, again, more motor, it has that precentral gyrus. The parietal lobe is more sensory, containing this postcentral gyrus. The occipital lobe. All right, so we know where the occipital bone is. We know the occipital lobe is going to be right above that. The main thing here is going to be the perception of vision. The visual cortex is where we actually are going to see what we see. It's actually going to allow us to see what we're looking at. The visual association area is going to give us the ability to interpret those visual signals, give it some type of meaning. And usually you're going to notice that association areas give things meaning. It actually kind of makes everything makes sense. So here we can see the posterior view again, just to kind of give you bearings. This was the cerebellum down below. This is the transverse fissure. This is now going to be the occipital lobe, all of this in the back. This area right here is the visual cortex, or we sometimes call that the primary visual area. And then this is the association area where it's given some type of meaning. That's going to be, sometimes we call that the secondary. The last two lobes are the temporal lobe. The main thing here I'd like you to know, the temporal lobe is hearing. Right? So right now, if you hear me on this recording, your temporal lobe is actually hearing it, but your association area for the temporal lobe is interpreting what you hear, right? So that the words make sense. Let's take a look at the temporal lobe. So again, if we go back to the side view, all of this here, I want you to be able to identify as the temporal lobe. And again, where the words that you're hearing right now, you're hearing them here along this part of the temporal lobe, but where it's given some type of meaning, right? We're trying to learn about the brain and you're trying to interpret all this information, is the association area. But the whole lobe is the temporal lobe. The next lobe, before I go to back to the slide, the notes, is going to be right in here, which is that lateral sulcus we had mentioned earlier. In order to see the next lobe, we actually have to drop the temporal lobe down and kind of raise the parietal and the frontal lobe up and kind of look into this fissure right over here. So here's a picture from our Martini book. You can see they actually did that. They actually lift up the frontal lobe and the parietal lobe a little bit, and they drop down the temporal lobe. And if we look in here, the next lobe is going to be the insula. And I took a picture. We can do the same thing on our model. Here's our temporal lobe, and I separated the other lobes above. This is the insula. Going back to our notes, the main thing I want you to know here is the insula is where we perceive taste and the perception of smell. Again, you can only see that insula if, say, on a practical exam or we were looking in the lab, we'd have to separate the temporal lobe below and then open up that and take a look in there. The last three structures of the cerebrum that we'd like to cover in this lab are structures that we can't see from the outside. We actually have to take the brain and we would have to slice it in that longitudinal fissure and open it up. So we'll be looking at a sagittal view. The three structures we're going to look at are the corpus callosum. We'll then take a look at the fornix, and then the septum pellucidum, which is part of the something called the lateral ventricles. So before I actually go, let's go to the slide, and we'll go back to the notes. So what we did here is we opened up the brain so that we're looking in where the longitudinal fissure is. The first structure, you follow my cursor around right like this, is this big C-shaped structure. This is known as the corpus callosum. This would actually contain fibers of neurons that would run across from this is the right side of the brain. I'm going too far. That was the right side of the brain over to the left side of the brain. The fibers are actually running across so that the right side of the brain can communicate with the left side of the brain. So that's an important function. But I also put in your notes, it covers something called the lateral ventricle. Inside the cerebrum here, it's not all solid. We actually have a cavity in there called the lateral ventricle. The cavity is actually C-shaped. So this structure, the corpus callosum, actually covers the top of that lateral ventricle. So I had put in your notes that it forms the roof of the lateral ventricle. The side wall of the lateral ventricle we could actually see right here. If we were doing dissection and if we took a probe and we just kind of poked through this membrane right over here, we would be in the cavity that's filled with fluid. So this wall right over here, we can call, you could say two things really. You could say it's the wall of the lateral ventricle, which I would be okay with. But the term that we use is the septum pellucidum. Right? Septum actually is a wall, and pellucidum is like a thin, kind of clear layer. You can almost look through it if you were doing real dissection. You can almost look through it, and if we poked our, like I said, a probe, a sharp probe, and kind of maybe rip this away, you would be able to see right into that lateral ventricle over there. I took a model and I actually turned it upside down, and I took out some of the lower parts here so we could actually look into these ventricles. So we could see here, this is the right lateral ventricle. Here's the left lateral ventricle. So now if I slice the brain away here in the middle in the longitudinal fissure, and right up here, this wall would be the septum pellucidum. That's where if I took my probe and I poked it in, we would now be in that lateral ventricle. So this is just a different way of looking at it. I would never ask you on a practical to identify it that way. I think that would be a little bit more difficult. But you should know this wall right over here, the septum pellucidum, is the wall of the lateral ventricle. There's another structure right here. This white tissue here is a bunch of neuron tracts that go to different parts of the brain, a little bit beyond the scope of this course. We're going to call this the fornix. This actually forms the floor of the lateral ventricle, so it's going to be on the underneath. So the three structures you're going to remember are the corpus callosum, septum pellucidum, and the fornix. And you could actually remember CSF, which is the abbreviation for cerebrospinal fluid. It helps you to remember that. And then in between here is where the cerebrospinal fluid would be. So you can use that. You can thank John from tutoring. He actually brought that up to me. It's a good way to remember it. Here's another model. Same thing, guys. It's just a different patient. Right here we can see the corpus callosum, septum pellucidum, and the white line over there, the fornix. No matter what model I gave you, even if they were different models, they would all look like that. And again, that was that kind of inferior view. So that completes the cerebrum. Let's take a look at the diencephalon. If you remember my introduction, the diencephalon is anything that has the structures with the name thalamus, hypothalamus, subthalamus, epithalamus, right? Anything that has thalamus is going to live here. We're going to do three of them. We're going to look at the thalamus, the hypothalamus, and the epithalamus. Before we go over these notes right here, let's just kind of do an overview. So we had saw before, this was the corpus callosum, septum pellucidum, and then this is the fornix. The first thing you should do, there's an almond-shaped structure right here. It just kind of looks just like an almond. This is going to be the thalamus. Use this as your reference point. Once you find the thalamus, anything below the thalamus is going to be the hypothalamus. So all of this is the hypothalamus. And then anything above the almond up over here is the epithalamus, right? Epi means above, hypo means below. Right? So go right to that almond, make that your starting point, and from there you can go up or down. So let's take a look at the thalamus. Right, so the thalamus, one of its major functions, it's a relay station. As sensory information is coming up from your body, again, like I said before, pain, touch, pressure, vibration, hot, cold, all the sensations that are coming up, you go to that postcentral gyrus in the cerebrum for interpretation. The signals are actually relayed at the thalamus. It's the thalamus that sends the signal to the appropriate lobe. So, for instance, visual signals go to the occipital lobe. Auditory signals go to the temporal lobe. Things like that. So all those signals go up to the through the thalamus as they're heading up to the cerebrum. The only sensation that is not relayed is the sense of smell. You don't have to know that for lab, but I do want you to know that for lecture. All of the signals going up to the cerebrum for interpretation are relayed through the thalamus, with the exception of smell. Smell goes directly to the temporal lobe and the insula for interpretation. It does not get relayed through the thalamus. Now, there's a thalamus on the left side and the thalamus on the right side. So the structure that connects the two is called the interthalamic adhesion. Right, another term that you may hear or read is known as the intermediate mass. It's going to connect the left and the right sides. So if we go back over here, find the almond at your thalamus, and this kind of darker area right over here, this has been cut, but this would go over to the other thalamus and connect the two, right? Everything's kind of connected. The left and right's always connected. The next area is the hypothalamus. This is really important. If you're taking AMP2 next semester, we study this in detail. It just controls so many things. One of the things it controls is the endocrine system. We'll learn that there's a gland hanging below it called the pituitary that's actually controlled by the hypothalamus, but it does a bunch of other things too. This is the part of our nervous system that regulates body temperature. Right? Your body temperature is always consistent at 98.6. When we get sick, it does allow us to get a fever. So it is very important controlling body temperature. If you feel hungry or thirsty, this is a big part of where those sensations come from. It controls our autonomic nervous system, the things that we don't control. We'll get more into that in lecture. And it, like I said before, it controls the endocrine system. The pituitary gland especially is under the control of the hypothalamus. Hanging down off the hypothalamus, there's a little body, a little projection called the mammillary body, which controls our eating. Again, like we said before, this is control of hunger. There's a stalk that goes down from the hypothalamus that the pituitary gland is going to hang off of, and we can see that pituitary gland on some of our models. So let's take a look at it. This whole area here is the hypothalamus. So again, it's below the almond, projecting down right over here, this little projection. This is the mammillary body. This controls our eating reflex. Now on this model, the pituitary isn't shown here. The pituitary stalk would be like right over here, and then the pituitary would be hanging down. This model does not have it, but that's where we would find it. But I want you to know for our class, this would be the hypothalamus. This would be the mammillary body. Let me just see if we have another. We have it on this model. So again, the hypothalamus is under the almond, which is here. This is the pituitary stalk. We call that the infundibulum. And this red structure right over here is part of the endocrine system. That is the pituitary gland. And if you remember when we studied bones, that little cup-shaped bone right there is the sella turcica, which is part of the sphenoid bone. Here we can see the mammillary body hanging down also on this view. Right? So this is a good description or a good picture of the structures of the hypothalamus. Let's go back to the notes. The last region of the diencephalon we're going to study is the epithalamus. So there's two things that we want to look at in the epithalamus. Again, epi means above. So if we go back to our almond, always make that your starting point. That's the thalamus. These blood vessels right here are known as a choroid plexus. We have a bunch of them in the brain and different parts of the body. Usually choroid plexus consists of capillaries that are leaky, and it's kind of an indication this is a place that we're going to be making some type of fluid. So these blood vessels are actually helpful in making cerebrospinal fluid. So again, when you just kind of identify all of these blood vessels in the epithalamus, this is going to be called the choroid plexus. If you follow this red line backwards, there's a little swelling posterior right here. This is called the pineal gland. This is important because it produces a hormone called melatonin. It helps to suppress the nervous system. It makes us drowsy. It comes out at night. You may know even people that take melatonin to help them sleep at night. They'll actually take it every night, and it just actually helps them fall asleep. But we do produce it naturally in the pineal gland. So with this next picture, I just did a blow up just to kind of go over all the different parts again as a review. Again, the almond is going to be the thalamus. Connecting the right and left thalamus is the interthalamic adhesion, also called the intermediate mass. Below the thalamus is the hypothalamus. On this model, we see the mammillary body, but we don't see the pituitary infundibulum and the pituitary gland itself. We saw that on the other model. Above the almond, right, above the thalamus, we're going to have the pineal gland here, secretes melatonin, and these blood vessels here that are going to help us to make cerebrospinal fluid called the choroid plexus. Right? That's going to be the choroid plexus. Again, anytime you hear choroid, think producing some type of fluid. Below the diencephalon, the next region of the brain we're going to look at is the brain stem. I like to think of this as the primitive brain. This is the brain that keeps us alive. It controls our heart rate, our breathing, blood pressure, just things that just keep us alive. Coughing, sneezing, vomiting. Very important part of the brain. So we're going to call this the brain stem. Like we said in the introduction, it consists of the midbrain, the pons, and the medulla oblongata. So let's take a look. So we saw before, all of this was the diencephalon. We just did that. All of this below now, guys, is going to be our brainstem. So this region up over here, let's say, pretty much all of this is the midbrain. This structure here, this reminds me of a walnut. Remember we had before our almond was the thalamus? The walnut is going to be the pons. It kind of look like a walnut. You'll be able to see that. And then down below that is this here is all going to be the medulla. Again, here's just a little bit of a blow up, a little bit closer. So let's take a look at, we're following our notes, the brainstem, the midbrain first. I put there also in your notes, we sometimes call it the mesencephalon. I usually refer to it as the brainstem. If we look here on the anterior aspect, I'm sorry, this is a posterior view. On the posterior aspect, on the midbrain, you're going to see 4 round structures here. Kind of literal translation would be 4 round bodies that we would call the corpora quadrigemina. This actually means 4 round bodies. The top 2 bodies are referred to as the superior colliculus. The bottom 2 are the inferior colliculus. The superior colliculus, again, if this is for lecture now, you need to know controls visual reflexes. Let's just say you had there was a big flash of light or something like that. It caused you to close your eyes and turn your head away. That would be mediated by the superior colliculus. The lower 2 bodies are the inferior colliculus, and they do the same thing as the superior colliculi, except they control auditory reflexes. So again, all 4 bodies you're going to call the corpora quadrigemina, which just means the 4 round bodies. The top 2 are the superior colliculi. The bottom 2 are the inferior colliculi. Now, I took a picture of the anterior aspect of the midbrain, and what we see here are some tracks. These are fibers that are running up. They're actually coming up from the spinal cord and heading up to the brain, like to the thalamus, which is ultimately going to relay to the cerebrum. So all these fibers running up, they're the ones that are bringing these sensations and the nerve transmissions from our spinal cord, from our lower body, up towards the brain. So we call these columns of fibers the cerebral peduncles, right? That's what I put in your notes, the cerebral peduncles. Again, you're only going to find them on the anterior aspect. The 4 round bodies, the corpora quadrigemina, are going to be posterior. We'll talk about this on a separate video, but the midbrain actually gives rise to the cranial nerves number 3 and 4. We'll talk about that separately. Below the midbrain, again, here just to show you here, this is the corpora quadrigemina from the back. You can actually see a little bit of those round bodies from the back right there. But below the midbrain now is going to be the pons. Like I said, it looks like a walnut. It has some important centers which control breathing and that type of thing, which is really, really important. All right, so here from a back view, we see the back of the walnut a little bit right here. And we could also see it on the front view. Again, here's all of this would be the pons, right? So you're going to find it right below the midbrain. Again, on a separate video, we'll talk about that. It originates cranial nerves 5, 6, 7, and 8. Below the pons, let's actually start with the sagittal section first. Below the pons, right, here's our walnut, below the pons, all of this now is going to be the medulla, and then that would be continuous with the spinal cord below. And so consider the medulla the lowest portion of the spinal cord. Again, a lot of centers in there for the control of heartbeat, a little bit of breathing, coughing, sneezing, vomiting, all these things that are important to keep us alive. We can see on the front of the medulla 2 columns coming up right over here. On the other side, on the right side, it will be over here. These are called the pyramidal tracts or the pyramids. They carry motor information that's coming down from the brain towards the spinal cord. So these they call the pyramids. They actually cross at some point here. We call that the decussation of the pyramids. And that's one of the reasons why when somebody has a stroke on the left side of their brain, the right side of their body is paralyzed, right? So that crossing is called a decussation. So these columns right here in the front is where the pyramids would be on the medulla. And here's the medulla from the front right here. Actually, this is the front picture right here. Okay. The last part of the brain is the cerebellum. So we had looked at the cerebellum a little bit earlier. We saw it was right below the occipital lobe, and it had the transverse fissure, and we saw that the tentorium cerebelli sat inside there. The tentorium cerebelli sat inside there. So generally the cerebellum controls all of our muscles. It controls balance, muscle tone, posture, equilibrium. So it has a lot of function. Originally, they thought there was a separate brain, and that's why they called it the little brain. That's what cerebellum means, but it's actually all part of our brain. So it has more motor control. If we were to look at the cerebellum, take it out, we'll see that just like the cerebrum, it has two hemispheres. There's a right cerebellar hemisphere, a left cerebellar hemisphere, and then connecting the two sides are these fibers that go across that is known as the vermis. Right? Vermis means worm-like, so kind of like these little structures of like little worms going across, or if you want to look at this whole thing here like a worm, that would be the vermis. Right? So all of that is the vermis. Now, if we slice the cerebellum, we'll actually see that the cerebellum also, just like the cerebrum, has a deep white matter, and then it's covered by gray matter over here. So there's like a cerebellar cortex. Remember we said cortex earlier is a covering, and then the white matter is underneath. So if you look at this, it's actually quite interesting. The white matter branches out like a tree, like the branches of a tree. So they decided to call these white matters that have a tree-like appearance the arbor vitae, right? Which actually means the tree of life. And then kind of like the leaves on the branches of the tree would be the gray matter, right? That's the cerebellar cortex. The last part of this video recording that we'd like to do is the ventricles. So we had mentioned earlier there's a series of these cavities called the ventricles. We're going to see that they make cerebrospinal fluid. That cerebrospinal fluid ultimately will go out and go into the subarachnoid space and go around the spinal cord and also go around the brain in the subarachnoid space of the brain. So we call this the ventricles. This is a picture from our Martini book, our textbook, showing the whole ventricular system that's been watermarked into the brain. So if you remember a little while ago when we looked at the cerebrum, we saw that structure was kind of C-shaped. It was called the corpus callosum. If you take a look now, underneath the corpus callosum is this C-shaped cavity right over here. This is called the lateral ventricle. This would be, in this picture here, the left lateral ventricle. It's in the left cerebral hemisphere. Then in the right cerebral hemisphere, they watermarked here the right lateral ventricle. As you can see, it's actually the same shape as the corpus callosum. And that's, again, because the corpus callosum is over the lateral ventricle. Now, the two lateral ventricles, they'll make cerebrospinal fluid. From there, the fluid is going to go through this little tube right over here. This is called the interventricular foramen. Right? Interventricular means between the ventricles, and we know from bones, foramen means a hole. The fluid will go through the interventricular foramen into this one slit-like narrow cavity called the third ventricle. In the third ventricle, more cerebrospinal fluid is made. From there, the fluid will go down this long tube right here. This is known as the cerebral aqueduct. Right? So an aqueduct just kind of carries water. And then the fourth ventricle is right here between the brainstem. Right? Here's the brainstem in the front. And then here's the cerebellum in the back. So this kind of triangular ventricle is the fourth ventricle. And then we'll see, guys, that the fourth ventricle is going to open up into the subarachnoid space and then the central canal from the spinal cord. So for lab, I need you to be able to identify these structures. I'll show you the model next. In lecture, however, I need you to know the flow, the sequence of flow. I usually have some type of sequential question. So I'll go over it again, just to kind of go over the sequence of flow. Cerebrospinal fluid is made in the two lateral ventricles. From there, the fluid flows through the interventricular foramen into the third ventricle, where more cerebrospinal fluid is made. From there, the fluid flows down the cerebral aqueduct to the fourth ventricle, where more cerebrospinal fluid is made. In the fourth ventricle, there are two apertures, which are openings, called lateral apertures. They send the fluid out into the subarachnoid space. There's one opening called the median aperture, and that's going to send the fluid right down here into the central canal, if you remember, of the spinal cord. So kind of review this picture, make sure you know where the structures are and the flow. The model we use is this model here. I can show you kind of a side view first, where we could see this is the left lateral ventricle here. Remember, it's always going to be the one that's C-shaped. And on the other side, here's the right lateral ventricle. You can't see it from here, but there's the interventricular foramen. It's going to be up over, I'll show you on the other slide. That's going to lead to the third ventricle. The third ventricle is only one ventricle, and if you look at it, it's more narrow and slit-like. Then that's going to lead from here to the cerebral aqueduct. And then the kind of reminds me of a triangle, I guess, a little bit. This triangular one is going to be the fourth ventricle. Here's a front view. This would be the lateral ventricle on the right. This would be the lateral ventricle on the left. And then here we can see right here is the interventricular foramen, and that's going to lead to the slit-like ventricle, the third ventricle. Here's a top view of the lateral ventricles. Again, this would be the left and the right. Actually, this would be the left. This would be the right lateral ventricle here. Down below, I labeled the apertures for you. So this is the fourth ventricle. Again, we can see the two lateral apertures. They're going to open up into the subarachnoid space. And right in the midline is going to be the aperture that opens up into the median. It's the median aperture that opens up into the central canal. So that'll conclude this video. The next video will be the anatomy of the eye, and then the anatomy of the ear. The next video after that will be the cranial nerves.